Now let's look at two more important macromolecules, proteins and nucleic acids. First of all, proteins. Proteins are made up of amino acids. Amino acids, there are 20 amino acids in nature, and the various combinations of amino acids will make up a given protein. So what makes the 20 amino acids unique are the different R groups that may be attached. On top, we see the amino acid tyrosine, and you can see the tyrosine group is substituted for the R group. Glutamine, we see the glutamine group is substituted for the R group. The amino group and the carboxyl group remain the same. So all amino acids have several things in common, and the only thing different is the R group that exists. So how do amino acids combine to form proteins? Again, remember the dehydration reactions we referred to in the formation of lipids and in the formation of carbohydrates. In the formation of those molecules, water was split out. When you look at one amino acid next to another amino acid, alanine next to glutamine, you can see that when the two come together, OH and an H are close by, thus splitting out, and the next to the right, again, OH and H split out to form water. The end result is a peptide bond that forms between the carbon and the nitrogen. So, in the formation of proteins, amino acids come together, water gets split out, and you form peptide bonds. The end result is a peptide. Here we're looking at 3 amino acids that have bound together. On the left side, you see an amino group. On the right side, you see a carboxyl group. But if you continue to extend this, you form, as you see towards the bottom, a long chain of amino acids we call a polypeptide or a protein. Now, the length of a protein molecule can be extremely long. We're looking at how a protein looks two-dimensionally and three-dimensionally. Now, first of all, as you can see, the structure of a protein is often looked at as a primary structure, which is dictated by the sequence of amino acids. I'm looking at alanine, leucine, serine, glutamine, and so on. This is my primary structure. My secondary structure results when the chain starts to fold. This becomes a secondary structure. There are several secondary structures, as you can see, beta, alpha helix, random coil. I'm not asking you to memorize those because the important thing is to understand how you go from a primary structure to a secondary structure, and finally the folding goes into a tertiary structure. Now, in a tertiary structure, what the amino acids have done, the polypeptides have done, is form a three-dimensional structure. The three-dimensional structure of a protein gives it characteristics. It's like taking something and carving out a unique shape. Every protein molecule will orient itself from a primary, secondary, and tertiary structure to have a unique shape, a unique three-dimensional shape that will give it functionality. This table shows you the different types of proteins and how they function. Basically, you can break down proteins into 2 types of functionalities. There are structural proteins, which make up muscles, which make up bones, which make up a variety of structures within an organism. And then you have functional proteins. Functional proteins would be things like enzymes, which carry out chemical reactions, antibodies, which are important in an organism's defense mechanisms, hormones, which are important in carrying out important messages, and so on. So look at this chart and try to get a reasonable understanding of the different types of proteins and their important roles in a biological system. Now we get down to the last of the macromolecules, nucleic acids. Now, nucleic acids we will deal with in a considerable detail as we move towards the genetic code and trying to understand how an organism stores information, how an organism dictates what activities are to be performed, and so on. But nucleic acids consist of DNA and RNA. The basic structure of the nucleic acid is the nucleotide. The nucleotide, as you can see on the left, is made up of a phosphate group, a sugar, and a base. There are 4 different types of nucleotides: adenine, guanine, cytosine, and thymine. This DNA molecule, as you can see, is a double-stranded structure, and each strand is made up of a phosphate sugar, phosphate sugar, phosphate sugar backbone, held together in the middle by hydrogen bonds between cytosine and glycine, adenine, and thymine. Therefore, ATGC, that is the binding characteristics of DNA. The DNA, of course, will form the double helix that all of you are familiar with. But for right now, we will leave any more discussion, we will defer any more discussion of DNA when we get into the genetic code. So let's summarize what we've learned in the last 2 lectures. Basically, we've learned that all of life is composed of 4 major macromolecules: carbohydrates, lipids, proteins, and nucleic acids. The table 3.4, as you can see, gives you a nice summary of the functionality of each of these molecules, and as we move along throughout, we will continue to expand and get a clear understanding as to how these molecules all work together to carry out the major characteristics of life.